everyone, and welcome to today's live broadcast, iPSC Derived Human Neurons, Introduction and Application, presented by Michael Hendrickson, a project manager at BrainCell Incorporated. I'm Susie Valdez of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by BrainCell. For more information about our sponsor, visit them on their site at www.brainxell.com. Before we begin, I want to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appear on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing this presentation, click on the Help Desk button located on the promotional board at the bottom center of your screen, or use that Ask a Question box and let us know you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located on the top right corner of your presentation window and follow the process for obtaining those credits. Without further ado, please join me in welcoming our presenter today, Michael Hendrickson. I will now turn the presentation over to him. Welcome, sir.
Thanks so much for that introduction, Susie, and welcome everyone. I hope that the conference is treating you well so far, even though we're just getting started, and that everybody enjoys the next couple of days. And as Susie mentioned, the title of my talk is iPSC Derived Human Neurons, Introduction and Application, and that's really what I'm going to try to do in this talk, is to provide you with a background in this technology, really from kind of a theoretical standpoint and a very practical standpoint, and then also look at some applications of the technology. And we'll be in particular discussing at the end of the talk a case study in which we looked at some drug screening paradigms using these cells. So a little bit of background on myself. I am, as Susie mentioned, a project manager and also an application scientist at BrainCell, and I've been with the company since its inception in 2015. I have a background in CNS drug development and also in neural development, so I have familiarity in this area. There's a picture of me there. There's action shot on the right side of the screen, although today I've cleverly hidden my bald head with this beautiful headset so you can all hear me well. I can be contacted at any time by email. I'm very responsive, so if you have any questions about this talk or request for the slides, which I think will be made available, or any other questions about the technological area, please email me at mhendrickson@BrainCell, and that will also be at the end of the presentation as well. So let's jump right in. I'm going to tell you about the topics we're going to cover today. I'm going to provide you with a little bit of a background about iPSC reprogramming and the nature of their pluripotency. This won't be too technical in this area because that's not our area or my area of expertise, but it'll give you kind of a general idea of how that technology works, and then how it fits into the main topic of discussion today, which is about neurons derived from those cells. And I'll talk again a little bit about how the differentiation is done. Again, this is going to be relatively general. There are a lot of protocols and publications you can look up if you have specific questions, but I'll give you a good idea of how that's typically done by our group and also in the field in general. And then we'll talk about how what types of neurons can be made using this approach, and discuss a bit about those different subtypes. And then for those of you who joined or attending this talk to learn a little bit about the nuts and bolts of actually using iPSC-derived neurons, I'm going to provide kind of a summary of how they're actually used in the lab, with some tips on use. And that should, I hope, maybe assuage some fears if people are thinking about getting into this area, and also provide you some ideas about how these need to be treated. These cells need to be treated a little bit differently from some of the, for example, cell lines or primary cells people might have worked with in the past. I'm going to talk a little bit about the application of these neurons. I'm going to kind of hit on multiple different topics, multiple different applications that we've worked on, and also our collaborators. And then, as I mentioned at the beginning, I'm going to take you through a high throughput screening campaign that we did using iPSC derived motor neurons, and that gives you a little bit of an idea of real world application of these cells and some of the challenges that we face and how we address those. So let's just jump right in with some background information here, talking about iPSC reprogramming, and then we'll talk about neuronal differentiation. So this slide provides a very general overview of this process. And really, one thing, if you're not familiar with this area, to remember is that iPSCs are derived from adults. So typically here, what's done is, in theory, any somatic cell can be used, but normally it's either a fibroblast or a blood cell, a PBMC, is harvested from an individual. So this can be an individual from age 1 to 100. It can be an individual who is not suffering from any disease, or an individual with basically any CNS or other disease. So once those cells are gathered, they're reprogrammed. So they're taken from that, say, fibroblast state into an iPSC state. And I perhaps didn't mention this in the talk, but iPSC stands for induced pluripotent stem cells. And so there are a number of ways to do this, but in general, the approach is to take these so-called Yamanaka factors and somehow get them expressed within the cell. As you can see here on the slide, there are a number of ways to do that. The most efficient ways, at least to date, are to use some sort of viral transduction. So Sendai virus is often used. You can also use an episomal approach or a direct introduction of mRNAs into the cells. So this is a very rapidly changing field, and so the technologies are coming out almost on a monthly basis. But in general, this approach is to get these these sort of master transcription factors into the cells that cause a reprogramming, really a reversion of the cells to a much more primitive state. And so the factors will change the entire transcriptomic profile of the cells and get them to that more primitive pluripotent state. So this is where the name comes from, right? You're inducing them into this pluripotent state. And from that point, you can take these cells and they can, they're proliferative, so more of them can be generated. They're like a like a true stem cell. They're very similar to an embryonic stem cell, but of course not derived from an embryo. And that the process of de-differentiation can be reversed, and that is we can take the cells and then differentiate them. So this talk will be entirely about differentiating them to ectoderm, and specifically to neuroectoderm, and then specifically to neuronal and glial subtypes. So here we're talking about differentiation to ectoderm and specifically to the CNS, and then specifically to neurons and glia. Slide presents a little bit of similar information, but it gives you an idea of the other areas in which these cells can be differentiated to. So in theory, iPSCs can be differentiated to any cell type in the body, any subcell type in the body. But of course, today we're going to be focused primarily on this lineage here, which is neuroectoderm down to neural cells. And the examples here are dopaminergic neurons and motor neurons. We'll talk in a minute about some of the other cell types that can be made. So this is the general scheme to get us to a neural fate. So the approach that our group uses, a general approach, is laid out in this schematic, in which we begin with an iPSC or an ESC culture, and those really, as I mentioned, can come from anywhere. And what is done typically here is really what's called recapitulation of development. So you're using various factors and culture techniques to pattern and drive the cells into more and more specific fates. So we begin what we've labeled here as an induction from the iPSC fate to a neuroepithelial progenitor fate. This is, it's somewhat similar to a neural stem cell fate, basically a neural specific but still high potency cell type. That cell type is then driven to a neuronal progenitor in our protocol, and so here we're producing specific neuronal progenitors. So you may have a motor neuron progenitor, you may have a glutamatergic neuron progenitor, and the way we approach this is we generate large numbers of those progenitors, which then get converted into early neurons. Those neurons can be differentiated and matured really into mature neurons, but for purposes of distribution to our colleagues and customers, we freeze them right at the stage where they're early neurons. And the bottom of the slide here just has a little bit of a description about the number of cells that are produced during this stage. And so you can start with a relatively small number of iPSCs and produce a relatively large number of neurons. So this is kind of a general approach that we use for all of our neuron types. But I wanted to give you kind of a sense of some of the details. So this is a published example of doing an iPSC differentiation to motor neurons. And you see there in the left where it says PSCs, that's just a pluripotent stem cell. So it could be an induced or an embryonic stem cell. And really here, this is kind of emphasizing the techniques to produce either rostral or caudal neuroepithelial progenitor cells, and then motor neurons with different phenotypes. And so this is something you can, people can certainly contact me offline if they'd like this publication or would request more details. But really the thing to keep in mind here is that the iPSCs are driven down. You're either ventralizing or you're dorsalizing or you're rostralizing or caudalizing to eventually get the cell population of interest, right? So you may be interested in cells from the forebrain or from the hindbrain or from the midbrain. And so all that needs to be taken into account at the beginning of the process to end up with neurons in the end that are the type that you wanted to produce, right? So these motor neurons in this situation are caudally organized, and so they're going to be spinal motor neurons. And so these neurons, as they are matured for 1 to 2 weeks in culture, begin to take on this beautiful morphology and expression of motor neuron markers. So this is just an example, but obviously we could step through multiple neuron types and provide the same kind of differentiation and maturation to produce very similar types of neurons. So these cells will have the classic neuronal morphology, and they will express the neural proteins of interest, which are detailed right below the figures there. And then really this is, again, to provide you an example of the morphology and the phenotype of these neurons at the end of the process, right? As I mentioned, this can be used, this technology can be used for many neuron types. And so we've already talked a little bit about spinal motor neurons, but of course, midbrain dopaminergic neurons can also be made. These were some of the earliest cells to be made using this kind of technology. We and others produce cortical GABAergic neurons, which have a variety of subtypes, including parvalbumin positive neurons and somatostatin positive neurons, and also, of course, cortical glutamatergic neurons. And these could be mixed together to produce, the 80-20 mixture that is seen in a in vivo condition. And these cells, using contemporary technology, can be generated with a very high overall neuronal purity, and they can also the neuronal subtype. So, for example, the percentage of all your neurons that are motor neurons, that purity can also be very high when the differentiations are done in a very controlled way. And as I hinted at in several slides ago, very large scales are possible. And we'll talk a little bit about that at the end of the presentation when we're focusing on the screening campaign that we did. The types I've talked about so far are more general types. We and others have begun to produce very specific types of neurons and astrocytes. So striatal medium spiny GABAergic neurons can be made, as well as layer-specific cortical neurons. So we can make very high purity layer 5 cortical neurons, which are involved in some diseases of the CNS. Hindbrain serotonergic neurons are another type that can be made that are not commonly encountered in this area. And as well, we can make cortical and spinal astrocytes. Again, many types are possible. So other groups produce sensory and peripheral neurons, and we and others are also working to produce microglia and other types of macroglia. So again, in theory, any neuron-specific type in the brain can be made. These are some examples of types that we've worked on in the past. So something that's very powerful as it relates to the use of iPSCs is the ability to generate neurons from patients. So as I mentioned before, the iPSCs can be generated basically from any individual, and regardless of age or disease status. And the same protocols that are used to make the normal, or wild type neurons can also be used from disease iPSCs, that is iPSCs from patients. And just an example here, you could make motor neurons from ALS lines that have a mutation for SOD1 or C9orf72. You can make glutamatergic neurons from Niemann-Pick lines, and you could make medium spiny neurons from Huntington's disease lines. And obviously the possibilities here are endless. And that's something that we and other groups have been working on. Just as kind of a practical guide, if you're out there and you're thinking, oh, this sounds kind of interesting, I'd like to do this, it can be, potentially a little bit daunting to figure out where to get the iPSCs. Potentially the lab down the hall has them, but also we've gotten them mostly from biobanks. And so I just wanted to mention a couple here that we've used in the past, and that would be NINDS and Cedars-Sinai. The NINDS biobank in particular has been very useful for us, and so I just wanted to give them another plug. And you can see the website there at the bottom of your screen. If you just Google NINDS biobank, you'll also find it very easily. So they have a wide range of iPSCs and fibroblasts and other cell types and tissue samples from a range of patients. And so that can be a very useful resource. Let's say you're at the point where you've produced some of these cells or you've purchased some of these cells and you're working with them actually in the laboratory. And so this is, can always be nice in these presentations, I think, to give them a little idea of actually, how it's done and what are the things I need to think about, especially for those of you who work at the bench. So I'll try to do that, and I'll try to provide some tips and some very practical advice as we go through this. So the first thing that you need to think about is, how you're going to culture these. And I'll just talk about from a commercial source, what people receive is, at least from us and from others, there's going to be a vial of neurons. And typically we're talking about single-digit millions of neurons in a certain fill volume. And of course, these are stored in the liquid nitrogen. Storage is important because these are overall very sensitive cells. So how you're going to plate them, what you're going to do really depends on the experiment at hand. So we have collaborators who plate these cells in very large wells because they need a lot of protein for a low abundant target. Of course, they could be plated, commonly in 96 well format for imaging, for cellular assays, for viability assays. That's a format we use very commonly, and even our guides talk about using that format. However, they can also be used in very high throughput formats. We've been successful in using them in 1536 well format, and some of our collaborators are even starting to do that, do some imaging and other screening in that format. And of course, they can be used in specialty formats such as multi-electrode array plates or other specialty chambers, microfluidic chambers or co-culture chambers. So I'll talk a little bit about some of those more esoteric applications later in the talk. And really, using these cells, if you follow certain guidelines, can actually be very easy because there isn't any passaging involved. So because they're terminally differentiated cells, you really just have to thaw them, count them, dilute them, and plate them out. And of course, wherever you plate them is where they're going to end up because they can't be lifted and passaged. Our medium that we use is presented here, and this is really just for reference. If you have this, you can get the slides later and have a look. This is also available on our website. A few things I want to point out is that it's best to have the medium freshly prepared. And in our protocol, we use a small amount of Geltrex or Matrigel dissolved into the media itself. And we found that provides better adherence for the neurons and also improves neuronal outgrowth as well, or neurite outgrowth, I should say. And in provided, at least from us, with our cells is a specialty supplement that helps with maturation. We recommend PDL-coated plates. We typically don't need or recommend using laminate PDL. We find provides a nice balance of giving good adherence of the cells, but also not necessarily encouraging them to migrate and clump together. So that's a common problem with when using these types of cells is to get a lot of clumping of the cells. And typically for most assays, you'd want nice, evenly distributed cells. And you can coat these yourselves, which is what we do, but they're also, PDL plates are also obviously commercially available. I'll just mention this quickly, but we and others have developed specialty supplements that assist with keeping neurons alive after thawing and also making sure that the progenitors convert, if there are any remaining late stage progenitors, that they all become neurons and that those neurons accelerate rapidly. Okay, so let's say you have these neurons in the laboratory and you want to actually use them. I'll just give you some really practical tips about using them and something to think about if you're designing an experiment. So as I mentioned, the neurons are relatively sensitive to temperature in the sense that they can't be stored in a -80, right? So they need to be stored either in an ultra low temperature freezer or in liquid nitrogen, which is commonly done. They can be shipped over short periods on dry ice for some of the neuron types. And of course, everyone says to keep the cells in the vapor phase, but we've had no problem storing them in the liquid phase. So if you're thawing cells, this is an important point I tell our collaborators and our customers, is to make sure that you don't over incubate the cells. So they are very sensitive to temperatures that are too hot. So you can kill them very quickly by going over 37 degrees. And so if you have any concerns or are nervous about it, I recommend to people that they turn their water bath down a little bit. And when you're thawing, if you remove when there's just a tiny bit of ice left, that's the perfect time, because by the time you get it into the hood, that last little bit will have melted and you're ready to go. As I mentioned, using them is relatively straightforward if you're cautious. And so the next step is simply to dilute the cells up, and you do that very slowly in a dropwise manner. You gently swirl, you don't shake the tube or triturate up and down. So we're trying to avoid osmotic shock, and as I mentioned in the last slide, also temperature shock, as well as physical shear of the cells. So if you treat them well at the beginning of your experiment, they'll perform really well at the end for you. You would count these cells in the same way that you would count really any cell type you're working with. There isn't much of a difference in doing the counting and the diluting up. Although one thing to keep in mind is that the viability will not be as great as you would see from some cell lines, for example. So typical viability is in the 80 to 50% range. If you see people talking about 95 or 99% viability, I would treat that with a grain of salt. That's a little suspect. because these cells are not the hardiest and they will not survive that freeze-thaw process without at least some cell death. And also, I would recommend that people note what the supplier provides as far as the number of cells that are either frozen or viable, because obviously there's a big difference there. In this example, we're plating the cells using a multi-channel pipette into a 96-well plate. Most times, people that use these cells are going to be using plates, and as a general rule of thumb, 1 million to 2 million cells per plate is kind of where you want to be when thinking about an experiment, for example. Obviously, depending on the assay, that will change a little bit, but that's a general rule of thumb if you're thinking about how many cells you might need for an experiment. And one little trick we use is that we typically will very gently plate into the wells and then leave that plate in the hood, let it sit, let the cells settle down evenly and attach, and you'll get a much more evenly distributed culture doing it that way. And you won't end up with cells in the middle of the well because the plate is being shaken. You also won't end up with temperature gradients causing cells to distribute elsewhere because they're in the incubator. Incubation is standard with these cells. There are some groups that use low oxygen incubators. We haven't found that that's necessary, although one challenge we do face in this area and others do is to maintain a very high humidity, and it's particularly problematic with long-term cultures in 384 or 1536 well format. So that's something to keep in mind, especially if you're going more than a few days in 1536 or more than a few weeks in 384, is that you're going to have that edge effect from evaporation. Another general rule of thumb working with these cells is the fewer changes you can get away with, fewer media changes you can get away with, the better. The neurons don't like to be shocked, and so changes just twice a week are perfectly sufficient for these cells. Just to recap some of these practical tips very quickly, we recommend using freshly prepared media to the recipe that we recommend or others, and our recipe is relatively standard. We also recommend using freshly coated plates that you either coat yourself or get from a commercial supplier. We've actually found that the ones we coat ourselves are a little bit better. And as I mentioned many times already, treat the cells very gently. If you treat them like an indestructible, immortalized tumor line, they won't be happy. If you treat them very gently, you're really going to get a beautiful culture out of them. And also remember that whatever vessel you plate them into is where you're going to need to run your experiment, right? So they can't be lifted and transferred without killing 95 or 99% of the cells. I'd like to talk a little bit about some applications of our neurons that our collaborators are working on, and these are really in no particular order and or no particular level of importance, but this gives you an idea of the kinds of things that can be done with our neurons, really across a range of experiments. So the first one I'll talk about, I mentioned a little bit earlier when referring to the specialty types of plates, is a multi-electrode array experiment, also called an MEA, or some people say MIA experiment. These are array plates that look like a typical microtiter plate, but they have embedded in the bottom of the well an electrode array. And that electrode array could vary by the supplier of these plates and their technology, and there are several around here. You can see that one of our collaborators works with Axion, obviously, and they're a reputable supplier for this kind of technology. In general, to run these kinds of experiments, the neurons are seated at very high density, and they may be cultured for actually relatively long periods of time. Although in several of our experiments, we've seen activity that's coming up at about a week. So some of these experiments can be run relatively early as well. Astrocytes in general improve the activity of these cultures, although they don't need to be run with astrocytes. And sort of leveraging the power of disease modeling, these kinds of experiments can, of course, be run with disease cells compared to normal cells. And that's something that people are looking at very closely. And so I just wanted to provide you with very little example data here. So this is just a quick single time point heat map from an MEA experiment that we did in-house. And you can see the white spots there are the actual electrodes where there's a high level of activity. And then on the right side of the slide, to give you an idea of what this looks like through time, is a time course of spike trains over 16 electrodes. You can see here, actually, if you kind of look at these traces, that there's activities kind of synchronized to some extent, whatever that is, every 30 seconds or something like that. So you can get really robust activity, and here we're talking about cultures that are 1 to 2 weeks old. So this is really looking more closely at the functional activity of the neurons rather than, say, protein expression or expression of a certain receptor. Also on the functional side, one area that can be studied is to look at the calcium influx of these cells. So you can load with a calcium dye, calcium indicator dye, and you can either do traditional microscopic imaging, or you can also do whole well imaging. So these are examples from the Hamamatsu instrument, looking at very long-term neuronal cultures for which calcium oscillations can be induced using several pharmacologic agents. Here we're looking really at network kind of level activity, right? You're looking at oscillatory activity within the cultures themselves. And this kind of activity potentially could be parsed for a disease versus normal condition. And these, building these kind of disease in a dish sort of systems, really here looking at overall network level activity. If this kind of experiment is done using imaging, you get images like this. I've got a nice video of this, which can't be used here, but to show sort of pulsing activity and oscillatory activity through time. So it can be done both as a kind of a homogeneous whole well collection of signal versus an imaging assay as well, using a calcium dye. So here we're looking at very long-term cultures, but acute changes that are not related to an oscillatory activity in the cultures can also be examined using various pharmacological agents. So this is just a really example of a quick experiment we ran with, I think these were maybe day 10 or day 12 neurons, applying different agents and looking for changes in the culture following electrical stimulation. Again, this is looking at calcium influx, but here you're looking at more of a real-time kind of pharmacologic response rather than a whole network activity. A really interesting application of these cells is high content imaging. They're beautiful cells, and actually they make for really good sources of imaging. They're not the easiest cells to image, but with a little bit of effort, the data that comes out can be really useful. So we have produced neurons that have a stably expressed GFP, and these cells can be cultured for 1 to 2 weeks, and various compounds could be applied as an example, and then images can be collected. So this is, I know this is difficult to see, but this is an example of kind of raw data you would get. And so potentially, a very large number of images can be collected. I think this group was collecting 4 images per well in a 384 well plate. So with one plate, you're quickly above 1,000 images. So obviously you need some computer assistance to do the analysis. And so here on the left side of the screen, you can see a control neuronal culture with nice, beautiful neurites extending from the cells. And on the right side, you can see a rotenone-treated culture. So you can use some high-content imaging software to help identify the cell bodies in these images. And then from there, the computer can build a trace of where the neurites are. Those neurites can then be quantified, so you can measure length and branch points, and all sorts of parameters to your heart's delight. And when this group did that, they were able to see not a huge difference between the number of neurons in each well, looking over time, with rote-known concentration versus normal or versus untreated. But the neurite length really provided a relatively large window between treated and non-treated, especially at the 48-hour time point. And so they were able to generate a really high Z prime for this assay. And they're currently taking this into even higher formats. So this has been one of the more interesting applications that one of our collaborators have looked at. Of course, neurotoxicity experiments can be done, again, looking at high content kind of formats. So this is just an example of an acetylcholinesterase assay that was done and converted from 384 to 1536 with very similar IC50 scores. The same group was also interested in looking at the neurons, human motor neurons compared to a human neuroblastoma cell line. And this is something I'll come back to before. The motor neurons are a much more relevant system, although the response they get is a little bit muted compared to the SHSY5Y cells. And you can see that by looking at the scale of these graphs. And so that's something that I think is important to think about, especially if you're looking at drug development, for really any situation in which you're using a model system, is to the extent that your response or whatever is being studied changes as you use more relevant systems. And so often, sadly, what we see, right, is that something that's studied in a cell line, maybe the response is less in a true neuron line or true primary or iPSC derived neurons, and that effect may fall off even further when a drug or treatment is taken into man. So that's always something to keep in mind with whatever model system you're working in. And finally, just to mention it, that transfection is certainly possible in these cells, and we can actually get, using lentivirus, relatively high levels of transfection. Non-viral methods are also possible, but the transfection efficiency is lower. This is something we're currently working on in collaboration with another company to try to get that number up. But it's a relatively straightforward procedure. It involves simply adding the virus at the start of the culture. So for the last part of the talk here, I'd like to walk everyone through a high throughput screening case study that we did. And again, for those of you who are interested in screening, this will be, I think, of particular interest, but also help to kind of highlight some of the challenges of working with these cells and how to address them. We did this in collaboration with the National Center for Advancing Translational Sciences, which is also called NCATS at the NIH. And we produced a very large number of neurons for this screen. And we looked at what, for us, was a large number of compounds. I know some groups have looked at more compounds, into the millions, but this was a large screen for us that we did from 5 libraries with a little help from a robot. And the overall rationale here, as I alluded to a little bit before, is that we encourage all of our collaborators, all of our customers to use the model system that's most relevant and feasible for the experiments they're currently doing. And so our view is that in an effort to kind of reduce the high rates of drug attrition, especially in the CNS area, which is highly costly, using systems like iPSC derived neurons as early in the process as possible can potentially help lower that rate of attrition and produce compounds that are more viable in the clinic. And so for this experiment, the model system, we're going to use our iPSC derived human motor neurons. We've already talked a little bit about those cells, that they have a complex morphology and they express the markers of interest. So the disease we're interested in screening for this, for compounds for, in this case, was spinal muscular atrophy, which is really a devastating childhood illness, and it can be fatal in type 1 patients, not fatal in type 2, and so on. The disease has relatively straightforward genetics, involves a mutation in the SMN1 gene, and most approaches to treat the disease have focused on increasing the amount of the SMN2 protein that's produced. So there's this potential rescue effect that can be achieved if the SMN2 level of protein can be enhanced. And so most approaches have focused on looking at the splice variants, changing the splice variants for the SMN2 protein. That's also the approach that we took. So we built a relatively complicated reporter system in which if the full-length SMN2 was expressed, we would also get the expression of our reporter, which in this case is nanoluciferase. So this system was a knock-in system at one allele at the endogenous site, so it really produces a physiologically relevant response. I just want to give a quick plug to Promega's nanoluciferase here because it's really performed very, very well in our hands. And it's more or less as advertised. So this is a very bright reporter. If anybody's looking to build their own reporter system, certainly have a look at it. It's very bright, and also I'll show a little bit data in a second that the false positive rate is in our hands was very, very low for nanoluciferase, which is not the case with all luciferases. So our reporter system responded to the positive controls that we were able to get our hands on. So these are both compounds in development to treat SMA. And the efficacy that we observed was about 50 to 60%. And we had a relatively low Z prime for a 96-well format, but because the system is very clean, we decided to move ahead and do the high throughput screening. So these details we don't have to get too involved with, but to give you a general sense, this is screening that's done over a relatively short period of time. But even over this period of just a few days, you know, the cells have already sent out pretty long neurites and have a morphology that's relatively mature. That doesn't mean the neurons are fully mature, but from a protein expression and a morphological standpoint, they have a reasonable level of maturity. So here you can see in the upper right there, just a quick image of these cells in the 1536 well format. And one thing to emphasize here is that these neurons were built from that reporter line. That reporter line was built on iPSCs from an SMA patient. So we're really looking at motor neurons with a reporter system from an SMA patient to have that proper genetic background. In this case, even though we would have liked to have used PDL-coded plates, we actually had to use uncoded plates, but because of the timeline of the experiment, which is relatively short, and because we were able to put a little matrigel into the seeding medium, we were able to deal with the uncoded plates. That's, of course, something we had tested beforehand. For these experiments, we used an approach called quantitative high-throughput screening. And so here, really, what you're doing is you're getting a dose-response curve for each compound. And that can be, for some of the libraries, up to 11 points, but also typically done at, say, 5 or 6 concentrations. It really helps you to rule out single-point deviations. Of course, it means running more wells. But for our system, in which the window was relatively small, this is really a very helpful approach to use, and it's an approach that NCATS uses quite often. So this is a little bit of an example data from the NPC-A library. A couple of things I want to point out here. As I mentioned, we didn't have a very large window using our positive control compound, but the system is very, very clean. And what that means is we were able to pluck out possible hits without worrying about pulling out a very large number of positive controls, especially when you would go in and remove potential outliers as these, because they were caused by a error in seeding at the so-called multi-drop error, right? Error seeding the plates initially. So as soon as we removed those artifacts, the number of compounds we had to deal with was relatively small. we're able to get overall very good CVs across the libraries, and this helped keeping the system clean, because again, here we're looking at a response of around 40%, a little bit under 40% for the positive control. So we needed a clean system in order to be able to see those possible hits. So even with these numbers, the Z prime is relatively low, but again, with a little bit of curation of the data, we were able to pull out some potentially interesting compounds. And so we had a low, so when we cast kind of a wide net, our hit rate was about 5%. When we looked through those hits using the quantitative data, we reduced that to about 0.5%. And then we were able to remove compounds that were true hits but uninteresting. So these would be compounds that inhibited protein breakdown. So in this case, obviously, we're looking for the increase in the level of a protein. So if you have compounds that are protease inhibitors, they're potentially will produce a physiological increase in that protein, but are not interesting, therapeutically. And unfortunately, we weren't able to confirm any hits from these screening, which is not terribly surprising because this is known to be a relatively difficult target. So that's just a quick example of a screening campaign that we did. And of course, people can contact me offline if you'd like more information about it. But I just wanted to before we get to any questions, to summarize what we talked about today. And so I really want to emphasize the fact that our view is that that iPSC-derived human neurons, in many cases, present a more physiologically relevant model systems, both for normal physiology and pathophysiology, especially compared to very unusual neuron-like cells such as PC12 cells or neuroblastoma cells. The other thing that's important is that these cells can be used much in the same way you would use those kinds of cells, but just a little bit of extra care and a little extra caution and some foresight need to be taken. But it's not something that should be overly intimidating. And these neurons are amenable to a range of applications. And here I've highlighted a few, but obviously most cell assays that can be done with these neurons. And again, they also are feasible to do large-scale screening with them. And so before we take any questions, I want to just thank our entire brain cell team who helped me put this all together, as well as our collaborators at various labs around the world, really. And I'll leave my contact information up here, and I think it's also available probably in the notes, in the lecture notes. But feel free to contact me really with any questions about the talk or this technological area. And if there are any questions, I think we can take them now.
Thank you, Michael, for that informative presentation. We will now start the live Q&A portion of our webinar. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your questions into the box that appear on the screen and click the Send button. We'll answer as many questions as we have time for. So let's get started and take a look at our incoming questions from our audience members. Our first question, Michael, is what kind of purity can be achieved and how is it measured?
Okay, that's a good question. That's a question we get often. And I think in one of the slides I just talked about high purity, right? So at least for the cells that we produce, and I can really only specifically speak to them, the overall neuronal purity that can be achieved can really be tremendously high. So they can be higher than 95%, and in many cases, they can be higher than 98 or 99%. So that's one way of looking at purity is out of all the cells, how many are neurons? But a more important way or an additional way you should think about purity is what percentage of those neurons are the subtype I'm interested in. And so that can range depending upon, whether we're talking about motor neurons or dopaminergic neurons and so on. So typically what we see is ranges from 50 to 80% for those subtype specific purity levels.
How long do the neurons need to grow before they can be used?
All right, so this is another good question. This is a question we get all the time. And I wish I could provide a simple answer, but it's really highly dependent on what you need to do. So you saw in my presentation that I presented data from as soon as 3 or 4 days after thawing. So in the screening application we did, we could get a nice response from our positive control at just day 4 after thawing. You also saw some data looking at network calcium oscillations that's taken from 7 weeks. So those really kind of provide bookends of the timeline needed for an experiment. So the more electrophysiological activity, the more network activity one needs, probably the longer one has to look. 7 weeks would sort of be at the outside edge of that. For most cell assays, we're dealing with looking at most receptors, protein levels, viability assays. We run most of those assays starting at day 7 and concluding at day 10 or day 14. So most of those are done within a couple weeks after thawing. But again, it really is highly dependent on the exact needs of your experiment.
Thank you, Michael. We have time for a couple more questions. And I want to remind audience members, whatever questions aren't answered today will be answered via email. Our next question from our audience member is, can they be used to make brain organoids or 3D cultures?
Yeah, so this aspect of the field is really coming on strong, especially the last 2 or 3 years. So we haven't done a lot of work in this area growing organoids or 3D cultures. The cells certainly can, as some of our collaborators are starting to look at this now. One of the reasons we haven't jumped into this area with both feet is that it can be very difficult to analyze those cells at the other end. It's certainly a very interesting technology, but adding the additional dimension when it comes to analysis, when it comes to consistency as well, adds a huge challenge that I think is potentially underappreciated by some people. So the cells certainly can be used to generate 3D cultures, but it's not something we have a lot of experience in, at least at this time.
Thank you for that. We have a final question. Can they be co-cultured with glia or non-neural cells?
Yeah, that's a good question. The straightforward answer is yes. So we and our collaborators often do cultures with neurons and astrocytes, and some of the groups we work with actually do cultures of, say, human motor neurons or human glutamatergic neurons with rat primary astrocytes. So certainly co-cultures with neurons and glia are possible. In addition, there are other possible combinations. So, for example, culturing motor neurons with skeletal muscle is certainly something that's been tried by several of our collaborators and is feasible as well. So those kinds of co-culture experiments, starting to build kind of organ on a chip type cultures are absolutely feasible. Yes.
Thank you, Michael. And would you like to provide any closing remarks for our audience before we close today?
I would just say, I encourage anybody to contact me. I'm obviously representing a commercial supplier, but we will help anybody with any questions they have and point them in the direction that will help them. So feel free to contact me with any questions and start thinking about whether these cells may, enhance the kind of research you're doing. Because we do feel really strongly that more relevant model systems, including iPSC-derived human neurons, will help to improve the translation of technologies, particularly in drug development, from model systems to the clinic, to the bedside.
Thank you so much, Michael, and thank you for this informative presentation and your important research. We also want to thank our sponsor, BrainCell, and I'd like to thank LabRoots for making today's educational webcast possible. Before we go, I want to thank everyone and let them know that today's webcast will be available for on-demand viewing through June 2018. You'll receive an email from LabRoots letting you know that the webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now, and thanks for joining us. We hope to see you again soon. Bye-bye.
Thank you, Susie.